Hi everyone, my name is Meagan Hoppe, and my project looked at the effects of a ketogenic diet on levels of prefrontal, cortical, and hippocampal metabolic and synapse-related proteins in rodents, in order to better understand the mechanisms by which dietary intervention can be useful to reduce the negative cognitive effects that are associated with aging. As the United States population of aged individuals is expected to double to 98 million in the next 50 years, it's vital to investigate approaches to mitigating cognitive decline. Aging is also associated with increased metabolic deficits, and this can contribute to cognitive difficulties as well. Ketogenic diets are composed of high fat, moderate protein, and low carbohydrates, which allows the body to transition from glucose metabolism to using an alternative pathway that uses ketone bodies. Previous studies in our lab have shown that a ketogenic diet can improve rodent performance and also changes levels of glucose and monocarboxylate levels in both regions, the hippocampus and the prefrontal cortex. We hypothesized that another mechanism that a ketogenic diet can restore cognitive function is through improvement of hippocampal synaptic function. To investigate our hypothesis, young and aged rats were either fed a ketogenic diet or a calorically and micronutrient matched control diet for 12 weeks and then sacrificed. The hippocampal tissue was purified by density centrifugation to extract the synaptosomes in order to look at synapse-related specific expression of proteins. And then this synaptosome was subjected to Western blotting to look at and quantify 10 synaptic protein levels with regards to neuronal signaling. Also, Western blotting was used to quantify age and diet-related changes of membrane and cytosolic levels of the glucose transporter GLUT4 in the prefrontal cortex and hippocampus of rats. In the GLUT4 study, we noticed very interesting results. GLUT4 is stored in the cytoplasm and mobilized to the membrane in response to insulin following meals. In the hippocampus, we observed no effect of diet. However, in the prefrontal cortex, when we compared the ratio of membrane to cytosolic GLUT4, we found significant interaction between age and diet, such that aged rats that were fed a controlled diet had significantly higher GLUT4 mobilization to the membrane than young rats. However, a ketogenic diet restored aged rats to their young counterparts. In contrast, in the hippocampus, our data showed that a ketogenic diet does not significantly alter synapse-related protein expression in any of the 10 synaptic proteins that we quantified. Overall, this showed us that while rats on a ketogenic diet display improved cognitive and affective outcomes, no protein levels were significantly altered by a ketogenic diet in the hippocampus synaptosome. However, GLUT4 insulin-dependent mobilization to the membrane seems to be elevated with age and restored with a ketogenic diet, suggesting that these diets work through restoration of metabolic impairments rather than through restoration of synaptic function in the hippocampus. Forward, we'd like to see if a ketogenic diet alters protein levels in a region-specific manner, so we'd like to look at these 10 protein levels in the prefrontal cortical synaptosomes. Also, since GLUT4 is mobilized in response to insulin, we'd like to look at a group of fasted rats to see if this impacts results. Thank you. Let me know if you have any questions.